Hi everyone, my name is Luke James and I work in the lab of Dr. Jada Lewis. Today I'll be talking to you about my research into the axolotl as a novel model for neurodegenerative disease. Many neurodegenerative diseases, including Alzheimer's disease, Parkinson's disease, ALS, and frontotemporal dementia, have been attributed to the abnormal aggregation of certain proteins. Therapeutics for these diseases have found limited success treating people in late stages of the disease and are primarily focused on halting disease progression. My research has focused on the utility of the axolotl, a highly regenerative salamander, to expand our understanding of the mechanisms associated with diseases and potential mechanisms to combat them. Pictured in Figure 1, the axolotl is an amphibian that, in contrast to humans, can regenerate damaged bodily structures, including portions of the brain. Utilizing the recently sequenced complete axolotl genome, I identified axolotl genes and proteins homologous to those implicated in a variety of diseases to assess the genetic capacity of the axolotl to develop neurodegenerative diseases. Here I present the analysis of some of those proteins. Microtubule-associated protein tau is thought to support microtubule stabilization, but can aggregate and form neurofibrillary tangles, one of the hallmark neuropathological markers of Alzheimer's disease. I identified the axolotl homolog of tau and annotated its internal domains, paying particular attention to the regions implicated in microtubule binding. 92% functional equivalence within the microtubule binding domain and increased levels of proline within the putative proline-rich domain suggest that axolotl tau may perform similar functions. I also looked at progranulin, a protein that plays a role in inflammation and healing processes. Mutations within the granulin gene cause autosomal dominant frontotemporal dementia. Using the NCBI conserved domain database, I identified 10.5 putative granulin subunits within axolotl progranulin, compared to 7.5 in humans. I also identified a homolog for axolotl precursor protein, which is cleaved to form A-beta peptides that accumulate and form plaques, the other hallmark of Alzheimer's. Further analysis revealed conservation of not only the cleavage sites within APP, but also the proteins that do the cleaving. The catalytic residues and active sites of presenilin-1 and beta-secretase were 100% conserved on the right of my poster, supporting the idea that APP is cleaved similarly in the axolotl. While neurodegenerative diseases have never been reported in the axolotl before, this analysis indicates that axolotls likely have the genetic capacity to be used as models and potentially get these diseases themselves. It is possible that axolotl neuroprotection and neuroregeneration combat the development of neurodegenerative diseases, and studying these pathways can actually aid us in designing novel treatments for neurodegenerative diseases. Thank you very much for listening.